Hello, everyone. I would like to welcome those who have just joined. Hi, Fortune. And hello, Abhishek. We'll be starting this session at the scheduled time. We are waiting for others to join as well. Meanwhile, we begin at one. I think we have met before as well. But Abhishek, Anshul, and Fortune, could you please tell us about your background and your research interest?
Hello, everyone. I
Hi,
am,
Fortune.
hello. I am Awala Fortune from Nigeria. I actually have a background in plant biosystematics. Um, in course of my research work during my doctoral studies, I actually focused more on molecular studies of plant species. And since then, I've been so interested in bioinformatics studies of related species, and my um, my involvement with um, Prime Biotech as I joined the Omis Logic program. I've been able to gain more knowledge on generally on other areas, including the um, knowledge on proteomics and analysis of uh, um, genomic data. So it has been quite interesting. Thank you.
Thank you, Fortune, for your introduction. We'll be starting the session in more 2 minutes as I see other participants joining too. Let's wait for them to join and then we'll begin. And meanwhile, we begin with today's session. It would be nice if you could please introduce yourself in the chat box. You can tell us about your educational or research background and also your research interests. Thank you. Hello, everyone. I would like to welcome you all to the second session on bioinformatics for infectious diseases. This program is dedicated to the study of viral diversity and its role in epidemic infectious diseases that keeps re-emerging, including zoonotic spillover, transmission between humans, and the process of viral and bacterial disease development. The participants will get a chance to learn about bioinformatics and analyze genomic data by applying various analysis approaches to study viral genomes. As a result, you will learn to understand relationships between viral strains and haplotypes, find differences in sequence data, and see the implications for drug and vaccine design. This program will provide opportunities to practice analyzing data to gain hands on experience with curated datasets from public domain collections guided by experts with bioinformatics experience and knowledge about virology. In the previous session, we discussed about next generation sequencing, viral genomes in host transcriptome. We went through an overview of NGS that included about reads, sequences, and file formats, learned about alignment, annotation, and non-mapped reads and alignment to databases of viral genomes. The 19 experience will almost certainly change the future of vaccine science. It shows how fast vaccine development can proceed when there is a true global emergency and sufficient resources. This wouldn't have been possible without a proper function healthcare system and industry. Today's session is in collaboration with Premas Life Sciences. Premas Life Sciences is engaged in delivering a high performance unit in genomics by establishing brands like Illumina and a few other niche technologies in the Indian market. Today, we'll discuss about NGS for infectious diseases, where we'll understand the workflow for designing your NGS Virology project, learn to process your samples, and generate NGS datasets. And then we'll also learn about virology public repositories and datasets where we'll find NGS data from public repositories and understand the virology datasets. Now, I would like to welcome our first speaker, Dr. Abhishek Pagela. He is currently working as a product manager and R&D associate in applied genomics domain at Primas Life Sciences Private Limited. He is primarily involved in developing NGS market in infectious diseases and other microbiology domains and is responsible for the development of novel NGS-based diagnostic solutions for a few important infectious diseases in India. We are pleased to have you with us today, sir. I would like to now pass on the stage to you. Over to you, sir.
Um, Thank you so very much, Sonalika, for the brief introduction, and a very good morning, a very good evening to all the participants over there. And so let me just share my screen. Right. Okay, I have also learned that probably you people have understood about basics of next generation sequencing in your previous training courses, but just wanted to start the presentation by having a little bit more about the impact of next generation sequencing and how this is very, very important in the current scenario, and how next generation sequencing can be applied for infectious diseases. So, we all are aware of the Human Genome Project, which cost, the cost of the project was about $3 billion, and it took 13 years to complete the project. And in that scenario, which is the case of when 2010, 2001, or 2002, Now, if you talk about the entire human genome project, sequencing of entire human genome, it can be done within few days, within probably a day or two, with the cost of about $1,000. So that is the advancement of technology we have, and this is because of the advent of next generation sequencing. And let me just give an example. This is so important that whenever you have to do a whole kind of a genome sequencing. So if you take an example of a chromosome, if you have to sequence a chromosome, for instance, what you should be doing essentially would be chopping off that DNA into smaller fragments, and those smaller fragments are then probably cloned in the vectors, and those vectors will be sequenced. And this was the technology which have been used in many for say, for example, Human Genome Project. And in that time, so I'm just putting an example of one chromosome. Say one chromosome, you will have 10,000 DNA fragments, and then you will have 10,000 clone vectors, and all of them have to be sequenced in a manner that you can probably go for 96 sample sequencing at a time, and to complete the entire sequence. Now, considering this particular task of sequencing the entire chromosome, it would probably take about 34 weeks if you have to go for Sanger sequencing. So in the Sanger sequencing, what exactly you're doing, you're basically sequencing all these different clones, and it would probably require a huge amount of money. So the impact of NGS is that, with advent of NGS, all these 10,000 fragments can be sequenced in a massively parallel sequencing manner together at the same time in a very short span of time and with a very, very limited cost. You can, so this is the power of NGS, where you can do so you have a power of scalability, throughput, and the cost. So this is the kind of impact we had because of advent of NGS. Now, before we really jump to the application of NGS for infectious diseases, let me just brush up your knowledge about next generation sequencing. So next generation sequencing, as we just discussed, there are multiple technologies, and among all of them, Illumina is the one which is highly efficient and the most adopted technology throughout the world. Now, how it works, probably you must have also learned this during the previous courses, but just let me go through once again, that you have your DNA which has to be sequenced. So what you do, you chop up the DNA and have adapters attached to it, and these particular fragments, they are attached to your flow cell. And in these, what will you do? You will have a bridge amplification, and then you will have, and what basically we do in PCR, that we take a DNA template and we try to have PCR primers against a specific gene and try to amplify that and go for sequencing. That is the case of Sanger sequencing. But in case of NGS, where you don't know the sequence of the genome, so you're chopping it off and trying to attach them to a flow cell and trying to clonally amplify all of them. And once they are amplified, they are being sequenced by a method called sequencing by synthesis. So, and as and when the differently fluorophore labeled nucleotides are being added, it is being captured in a high throughput manner. And that is how, you consider one particular spot and this particular cluster. So in one shot, you have a G. In the secondly, what is added is C, then followed by T, G, and A. And that's how the entire DNA is being called for in one particular cluster. Likewise, you have millions of clusters, and you tend to get a very high throughput data about the genome sequencing or whatever application you are probably interested in. So now coming to the applications, when we talk about NGS, we have scalability. So you can basically try to go for whole genome sequencing. One can go for exome sequencing, and then you can go for Amplicon sequencing. We'll try to cover some of this portion with respect to infectious diseases. While Sanger sequencing, you probably go for single gene, and you have a low scalability for that. Now, coming to infectious diseases. So when we say infectious diseases, it's just not only confined to the viruses. You have plenty of bacteria, fungi, parasites. They all are cause. Many of them are causative agent of infectious as infectious diseases. And if you look at this particular chart, we have had very tremendously affect what do you call the infectious diseases which have affected the human civilization very significantly. And one of them we are experiencing right now, which is COVID-19. And this is one thing which is this is something which probably brings the entire focus of NGS towards the mainstream science. Now, infectious diseases they can be passed from person to person, and some are transmitted by insects or animal bites, and then some are because of contaminated foods, and there are many more ways one can get infectious diseases. So, what is important here is to understand that how one can use next generation sequencing for infectious disease diagnosis and surveillance. So, this is a very broad area of research where the idea is there can be three different ideas. One is the pathogen identification. At times what happens that, when we work with the clinical samples, that time it is difficult to culture organisms directly from the clinical sample. And that creates problem, and that becomes, rate limiting step for a very accurate and early diagnosis of the disease. So for that matter, it is very important that NGS can be applied there. And of course, what is another important point is that, when we have pathogen identified, or when we have pathogen which we have recovered from the clinical sample, one can basically look for what kind of resistant mutations are there for antibiotics, and what kind of toxins or virulence genes are associated with that particular organism, if the organism is found to be highly virulent. Other than that, there's a whole range of host response which can be studied. It's not only the pathogen which one can try to study, but of course, host response can also be studied. So, which is a different arm altogether. Coming to a very important area, which is the surveillance, and which comes under epidemiology. So there have been various hospital outbreaks of various infectious diseases, and there are many outbreaks not only in hospitals but in community also. And so NGS has played a very important role with respect to outbreak investigations also. And that's how strain identification is very important when we try to do outbreak investigation. And another one is the community structure, when we try to find out what is the microbiome of a given sample. Say, for example, now there have been multiple studies which are linking gut microbiome dysbiosis with various diseases. So there is another area where NGS has got an application. So today, what we are going to probably discuss is more of maybe more towards pathogen diagnosis and understanding the resistant mutations, and then further how NGS can be applied for microbiome analysis. Now, there are two very broad category of the methodology one basically go for while applying next generation sequencing for infectious diseases. One is targeted amplicon sequencing, and one is whole genome sequencing. So let me probably revise you about this concept called 16S ribosomal RNA. I'm sure many of you are aware that 16S ribosomal RNA gene has been classically used throughout the world for bacterial identification. And similar to 16S ribosomal RNA gene, there is another gene called internal transcript spacer region, which is called ITS, and that has also been classically used throughout the world for fungal identification. Okay, so these are the genes which have been classically used. Now, the approach goes this way, that when so this targeted amplicon sequencing, basically, so what we do in this, we know what is a target. That means what we can go for, we will try to go for 16S ribosomal RNA based approach for sequencing, or maybe IT's based approach for sequencing. So in these situations, say for example, if you try to imagine a sample, which is say for example, a sputum sample, from there you have isolated total DNA, and then try to amplify specifically what all bacteria are there in the sputum. So this becomes very important because you are essentially trying to enrich the bacterial DNA while leaving behind the host DNA. So this is an advantage. So what we do, we try to amplify all the possible bacteria which are there in the sputum sample and try to sequence them and find out what all bacteria are present in the sputum sample. This is just one example. I'm just putting up that way. Another approach is whole genome sequencing, where you have possibly isolated the bacteria or fungi or virus, or the virus in the cell culture. So you want to know the whole genome sequence, so that in that situation, the strategy would be different, that you would go for a different strategy. So we'll see how these different approaches are applied in case of infectious diseases. Now, the very first disease which we are going to talk about is, of course, COVID-19, because that has changed the way we'll all live, and probably it has affected all of us in some or other manner, and this is the timeline what we could see, that this has become such an important disease, particularly in India, that we have had the very high load of COVID-19 patients in India. And this is something which is very, very important, in a way that because of the COVID-19, every other person is aware of RT-PCR test, and every other person is now aware of whole genome sequencing or genomic surveillance of SARS-CoV-2. Because of that, the next generation sequencing has been brought to a center stage, where it is helping the infectious disease specialist or the policymakers to make the decisions, And so in these particular disease, I'm trying to put up certain statistics of India, where we had very high number of cases of coronavirus during say May 2021, and then again we had a peak of Omicron probably in January, which is subsided now. So the point is that COVID-19 as a disease is highly dynamic, and not only dynamic, it is evolving. So what we have been essentially doing is to diagnose the disease. And how do we diagnose the disease? By doing COVID-19 RT-PCR test. Okay, so there is a difference between diagnosing a disease and doing a genomic surveillance, because by diagnosing the disease, you could say that, okay, there is a COVID-19, that the disease is there, but then you might not be able to tell what variant is causing the disease. So when you talk about all sorts of variants, starting from alpha, beta, gamma, delta, all this information is possible through genomic surveillance. That means through genome sequencing of the virus. So here is the era of the genomic surveillance of SARS-CoV-2 virus. This is very important, because the genomic surveillance is why this is very important, because it can help us to track the transmission routes of infectious disease globally. And now we know how the disease started and how it is spreaded. And we will be able to we have understood how rapidly the virus is mutating. And then there are certain mutants there are certain variants which are probably more they might make vaccines less effective, and then they probably evade diagnose the test also. So this is very important, that we should know the entire genome sequence of the virus, and then so that one can identify which variants are there, and based on the variants one can have proper strategy for control and containment of the disease. And with these respect, we have so this is a classical example of how the workflow goes for genomic surveillance, that you basically isolate RNA from the patient sample. And what classically is being done for COVID-19 diagnosis is that qPCR detection and the report. But in case of genomic surveillance, we go for RT-PCR, PCR, and then go for PCR amplification, library preparation, Illumina sequencing, sequence analysis, which will include all your different strategies of phylogenetics, phylodynamics, and variant classification. So that is how we identify what variants are there, what lineages of viruses are there, and this is a classical workflow for a genomic surveillance of SARS-CoV-2 virus. Now, in case of genomic surveillance of SARS-CoV-2 virus, Illumina has been at forefront throughout from the beginning to today. To date, we have been probably serving the entire genomic fraternity for genome sequencing of the SARS-CoV-2 virus, and we have a couple of solutions for sequencing of SARS-CoV-2, where we have shotgun metagenome sequencing, enrichment-based panels, and then the Amplicon-based panels. And in Amplicon-based panel, we have a COVID-seq test. This is a test which has been used in many of the genomic lab for the genomic surveillance of SARS-CoV-2 virus. And this is a COVID-seq test which can actually enable detection and sequencing of SARS-CoV-2 in research setting. And we have a couple of sequencer where it can be run in it can be run in all these possible sequencing platforms what we have. And the workflow goes this way, that you have RNA which is extracted, and then we have a 98 amplicon. We have to amplify 98 amplicons using different sets of primers. So there'll be COVID-seq primer pool 1 and primer pool 2. These two in total will amplify 98 amplicons, and that will be sequenced, and then we get the complete genome sequence of the virus. And then based on the analysis, based on the COVID lineage app, we get to know what kind of which lineage of the virus is there, which variant is there, so that all can be done through this particular end-to-end solution. So this is exactly it goes, that we collect the sample, and then here's sample preparation, library preparation, sequencing, analysis, and then report. So this as I said, this is end-to-end solution for the genomic surveillance of coronavirus, for the coronavirus. And now we have been seeing that there have been many variants which are popping up and then going down and stuff like that. That you see there have been variants like alpha, beta, gamma, delta, as we discussed. And then recently we had Omicron, which was like we had huge number of cases in India, and suddenly it has just subsided now. But that's not the end of the story. Because the coronavirus is having such kind of a genetic makeup that it will keep mutating, and then probably we'll have new mutants arising in future. And this is on the right hand side. You would see a news published very recently in the New York, which talks about that there is a mysterious coronavirus which was found in New York City sewage surveillance, and they found that it has a huge set of mutations, and those are very unusual mutations. So you see that the things are not really have gone down, and you never know when the virus will come back with a new set of variants, and that becomes very, very important, that we should keep our guard. We should not, we should always be equipped, should be prepared for genomic surveillance of SARS-CoV-2 virus in future. And in this particular aspect, Illumina COVID-seq test remained the most important tool for genomic surveillance of SARS-CoV-2 virus. Now, in India, we have had a consortium of Indian SARS-CoV-2 Genomic Consortium, where we have a set of laboratories from India which are involved in SARS-CoV-2 genome sequencing, and and if you look at the entire scenario of SARS-CoV-2 genome sequencing, we will see that in India, approximately 93% of sequencing is done on Illumina platform, and because of its accuracy and its wide applicability. Now coming to, and once we get the data, has to be uploaded to the GSET site, and probably you must be aware of this site. And in case of COVID-seq assay, which is from Illumina, we have an option where the data is updated into GSAT, and a data analysis also is coupled with GSAT. Probably Raja, the next speaker, would be able to talk more about the analysis part. Now, this is all about the SARS-CoV-2 virus. Now, and there have been multiple viruses in past where the NGS has played a very, very crucial role while investigating the outbreak. And Ebola is one of them. Probably many of you are aware of that. Now, moving on from virus to bacteria, I think this is another important area where which has got a lot of attention in the recent time, especially tuberculosis, because what has happened that because of the COVID-19, the entire fight against tuberculosis has got a setback because of a lot of lockdowns and stuff like that. So, but tuberculosis has remained one of the major important disease as far as developing world is concerned. And what we see that tuberculosis, obviously we're aware that it is caused by Microbiome tuberculosis, and most of the cases are you have pulmonary tuberculosis. But nowadays what you see that there are cases of extrapulmonary tuberculosis as well, which include intestine, meninges, lymph nodes, bone, joints, and kidneys and skin. And there are there's so high number of cases, and there's so high mortality associated with disease. And because of the importance of this disease, WHO has set up a mandate to end tuberculosis by 2035. And especially to reduce the burden from, at least not complete elimination, at least to 95% reduction in TB death by 2035. Now, what is important, tuberculosis has been there for ages. What has become more important that, what we have probably more problematic situation in India, that we have about more than 24 lakh tuberculosis cases. And we probably report more than 25% of global TB incidences in the world. Now, this has been there in India for so many years, and then we have had multiple national programs to eliminate tuberculosis, and there are so many stakeholders involved in that program. But the problem is again compounded by emergence of multi-drug resistant and extensively drug resistant tuberculosis, wherein the Mycobacterium tuberculosis is resistant towards a few set of drugs, and that becomes difficult, and then that becomes very difficult to cure disease. And if you look at the kind of load we have for as far as MTB or as far as Mycobacterium the MDR TB or XDR TB is concerned, you could see that we have very high number of cases of XDR TB and then MDR TB as well. So that becomes problematic. Now, so because of this, what is important now, that we need to diagnose the disease in a very early stage, or we need to diagnose the MDR TB and XDR TB very accurately, so that one can actually provide better solution, one can prevent the disease and control the entire cascade. And now, what is most important? TB diagnosis and diagnosis of drug resistance in Mycobacterium tuberculosis. These two things are very, very important. Now, look at the level of what we do have it in India, that we have primary healthcare, secondary healthcare, and tertiary healthcare setups. And in order to identify or diagnose the disease using sputum smear microscopy, which is limited to primary healthcare, and one has to go for test for diagnosis of MDR and XDR TB, then it is more restricted towards tertiary care center, where you have huge infrastructure and which requires kind of a biosafety level, which is required for tuberculosis. So we have a problem with respect to diagnosis of disease, and with respect to identifying the drug-resistant tuberculosis, the Mycobacterium tuberculosis bacteria. So how this can be further? This is one of the problem what I just mentioned. Another problem. So I'm trying to portray that there are few problems and how NGS can really help in the decisive fight against tuberculosis. So you can see what I have mentioned here, that tuberculosis, you have an evolving molecular epidemiology of Mycobacterium tuberculosis. So basically, Mycobacterium tuberculosis has originated from, say, Africa. It has evolved from Africa, and then it had spreaded to, so it has formed multiple lineages, and these lineages are like kind of a set of very specific genotypes. So these lineages of MTB, so these lineages are not different species. So you have same species of Mycobacterium tuberculosis, but they are having different lineages. And these lineages, you have, say, a couple of lineages. Now this lineage 2 is more virulent with high mortality. It has high mutation rate. It to acquire drug resistance very fast. So this is a Beijing lineage, which is more predominantly found in China. Likewise, we have a lineage 4, which is more likely to have consolidation in the lungs. And then there's a lineage 1, which is more likely to present with bilateral cavitation and lower rates of TB transmission. And if you look at all these things, you would wonder that even in India, if you look at in India, we have lineage 1, which is predominant in South India, and lineage 3 predominant in North India. Now, this is very important that, all these lineages are very, very important to identify. And if you look at in India, we have set of lineages. So you look at in North India, we have a different lineages. South India, we have a different lineages compared to Eastern and Western. So these identification of these lineages is very important, so that one can have a necessary intervention for control of tuberculosis. And all these lineage identification is classically be done by a genotyping method, which is very cumbersome method and which can only be performed at tertiary care centers where you have a huge infrastructure. And this is called polygotyping. And then you can also use some other dot blot assay for that. So now how these things can be really, taken up by next generation sequencing. So in India, what we see that other than these problems, tuberculosis, we have another issue that we have we are aware of the fact that tuberculosis is caused by Mycobacterium tuberculosis. But there are some non-tuberculous mycobacteria are also there. Non-tuberculous mycobacteria are very important because they are often being seen as a co-infective agent with MTB. So why this is important? These NTMs are important because they are very resistant. They are highly resistant towards all the conventional anti-tubercular drugs. So if a patient has an infection of Mycobacterium tuberculosis and a co-infection of NTM, and if that patient is being treated for Mycobacterium drugs, the patient will not respond, and it will have the symptoms and the disease persistent. In that situation, it becomes very important that we need to identify non-tuberculous mycobacteria as well, in addition to the main organism, that is Mycobacterium tuberculosis. Now, this NTM is being identified using line probe assay. A line probe assay is kind of a very specialized molecular assay which identify different species of Mycobacterium tuberculosis. Now, what has been seen that in case of in India, what we see by applying whole genome sequencing, people have identified that the co-infection rate of NTM with M tuberculosis is about 20%. And this was the rate which was never detected before, because initially the identification of NTM was restricted towards few centers because these techniques are specialized now. But because of whole genome sequencing, the Illumina-based sequencing, NTMs are also being identified, and because of that, the people are able to get a more accurate treatment using this kind of an approach. Okay. Now we are aware of a fact that that MDR tuberculosis, that is multi-drug resistant tuberculosis, is because of the mutations in the drug target for rifampicin and isoniazid. But in India, or probably I'm sure it must be happening in many other parts of the developing world, we do see various novel mutations which are there in case of MTB, and such mutations cannot be detected by any of the methods. For that, we need to go for next generation sequencing, Illumina-based next generation sequencing. And so in this situation, what we offer, next generation sequencing offers that one can go for laboratory diagnosis of tuberculosis bacteria and non-tuberculosis mycobacterium. So classically, this is being done by doing smear staining or by doing Biotech MGIT culturing or by line probe assay. Now, another important point is that we should know whether a given bacteria. You should also understand that Mycobacterium tuberculosis culturing is very fastidious. It takes weeks to grow, and once it takes weeks to grow, then once it is grown, then it is being subjected to physical drug testing to identify what are the drug resistant mutations in that bacteria. That again takes few weeks to get the results. So in that situation, the entire treatment gets delayed, and that basically helps the bacteria to probably spread more efficiently. So this is another issue. And for that also, in classical setting where you don't have NGS, you have to use Biotech MGIT or some other methodology. And I just mentioned you about that how lineages are important in case of MTB, and how spoligotyping. This is classically done by spoligotyping, but by using NGS assay, one can do all these different sets of analysis in one go, and one get a comprehensive picture of the entire disease spectrum, so that a decision can be made very, very promptly by clinician. Okay, now how do we go about that? So for that, we have a assay called Diplex MYC TB. So this is a targeted NGS based assay. So targeted NGS based assay, that means we have what we have here. We have a panel of so in this particular assay, it targets it identifies mutation in different drug resistant genes. For example, rifampicin, isoniazid, and ethambutol. So these are first line antibiotics. Then you have second line antibiotics, and even the new antibiotics which are like bedaquiline or linezolid. So through this particular assay, we are able to identify mutations in all these drug targets. Other than this, there's a gene called HSP65. So this is used for species identification. That means that one can identify Mycobacterium tuberculosis. Not only this, but one can also identify the NTMs, the non-tuberculous mycobacterium. Okay. So in addition to this, I mentioned you about identifying lineages. So there is a panel for spoligotyping that identifies lineages, and there's phyloSNPs which is another genotyping method. So through this particular comprehensive approach, we can simultaneously predict the resistance to 15 anti-TB drugs, and also we can identify different NTMs and MTB, and also identify lineages. And this is a very interesting solution. There the total turnaround time is 48 hours. And most importantly, it can be directly applied to sputum. So you don't have to culture the bacteria and isolate DNA and use this assay. This assay can be directly applied to sputum. Okay, and you can have a possibility of having 384 samples in one go. So this is how it goes. And so you have a sample preparation. This is an end-to-end solution what we have here. So you are aware of the fact that there are a lot of NGS assays where you have to you have to put a lot of efforts to for bioinformatic analysis, but in this situation, when you have DNA which is being subjected towards library prep for this MYC-TB, we have a web-based analysis platform where at the end of at the end of the test you get the entire report. I am showing you a sample report that you get a patient name and the sample type, and on which sequencer this sample was run and what was the method. Say, for example, this is whole genome sequencing, but you can probably talk about, say, duplex MIC-TB also. And then it will talk about lineages, which lineage is that. And then it will tell you this is resistant towards isoniazid rifampicin and what kind of a mutation is there. That kind of information also one can get. So this is something which is quite, a very end-to-end solution as far as Mycobacterium tuberculosis disease is concerned. So this way, we will have a very comprehensive picture for disease diagnosis, for understanding the epidemiology of the disease. Now, coming to another important area, where we have this called a silent pandemic. We have been listening. We've been talking about SARS-CoV-2 because this is something which is everybody is aware of. And antimicrobial resistance is a silent pandemic because this is, and it is so much so that it is, WHO has identified it as one of the biggest threat to the global health. And it is going to have a tremendous impact in the years to come. You can just, imagine a situation in 2050 where up to 10 million deaths will be there per year if nothing is done to contain the AMR problem. And it is going to have a significant effect, so much so that it is going to affect the GDP of many countries, and the cost involved would be tremendous. So now let me tell you where are the problems. You are aware of the fact that one try to identify AMR in a clinical setting. Say, for example, you're working on a bloodstream infection and probably working on Staphylococcus aureus, or maybe some streptococcus, or maybe Klebsiella, or any other organism, and then trying to, probably you have isolated those bacteria from the blood or from clinical sample, and then trying to see whether they are having they are having any resistance towards antibacterial drug. So what we are essentially doing, we are basically at the moment looking at only one arm of the entire interwoven network of AMR problem. So what we basically are doing, we are basically in a hospital setting, we are trying to identify whether we have any of these in AMR or antimicrobial resistant bacteria in our bodies, especially in clinical samples. And now, so this is one of the aspects which we are probably trying to look at, but the picture is quite wide and it's bigger. Why? Because the problem is that we have irrational use of antibiotics, especially for poultry, for animal farming. And because of this, we are probably trying to propagate or probably induce AMR into the bacteria or the microorganisms which are there in their gut. And these microorganisms directly or indirectly we consume. And that basically finally leads to having the resistant bacteria. Not only about these arm of the AMR, but then you do see irrational use of various antimicrobial agents in environment, and probably in aquaculture also you do see that. And there are multiple ways you would see that the irrational use of antibiotics and antimicrobial agents. So what becomes important that, it is very important not only to screen or do a surveillance of AMR in human population, but also to do a surveillance in the environment. And here comes the scope of next generation sequencing. Because, you are probably, if some of you are aware of the fact that, antimicrobial susceptibility testing is a very cumbersome process. And not for every bacteria one try to go for physical drug testing. So in that situation, when you have thousands of bacteria which are isolated from environmental samples, you would never know whether they are really going to harbor the antimicrobial resistant genes or not, or whether it is really practically feasible to go for physical drug testing of so many bacteria from environmental samples. So in that situation, NGS comes into action, and there NGS can help us in understanding the AMR spectrum. So there can be three different scenarios. One scenario is that you have a patient who has probably a kind of a bacteria and in his blood or in his blood, and then somehow the patient is not responding to the empirical antibacterial therapy. So what you do, you essentially try to go for genome sequencing and try to identify what kind of mutations are there. Obviously you try to go for physical drug testing, but still, one can go for whole genome sequencing to identify what kind of antimicrobial resistant genes are there. And so that is one thing. Now, when you do not really have a strain which is isolated from the clinical sample, what you can go for metagenomic analysis from clinical sample directly. So this way you would be able to identify the pathogen directly from the clinical sample. Then at the same time, you would be able to also identify if there are any antimicrobial resistant genes are there or not. And then that will help us to understand what kind of a resistant pattern is there. Now, what is most important is the environmental samples, where you cannot do a physical drug testing for every bacteria you isolate or every organism you isolate. Here it becomes very, very important, because in these environmental samples, we can apply next generation sequencing based on the metagenomic analysis, and one can identify whether there is a presence of antimicrobial resistant genes in the effluents of human or agriculture activities or wastewater, of the cities. And if there are, then one can try to locate and pinpoint the source of such kind of antimicrobial resistant genes coming from which particular settings, and then try to assess the risk of these ARG getting transferred from environmental commensals to the pathogens. So this becomes very, very important. And then so in this regard, we have a NGS can have a comprehensive genomic solution to track the antimicrobial resistant through shotgun metagenomic analysis or whole genome sequencing. In addition to this, in we have a AMR panel with Illumina, where we provide a comprehensive solution for more than 28 different drugs and 490 mutations for different drug resistant mutations. So that is a comprehensive solution what we have, and which can be directly applied to any kind of samples which one can go for. And that's how the entire. So once we know where the resistance is coming, then one can actually take a decisive step to contain the resistance, and that's how we stop the for the spread of the AMR. So this is about AMR. Now, coming to the last portion of my discussion today is about the microbiome. Microbiome has largely been, if you look at microbiome, is now linked with probably many disorders, or maybe many different conditions, starting from depression to allergy to neurodegenerative disorders, obesity, diabetes, and there are many more. Especially if you look at gut, you're aware of inflammatory bowel disease, where the gut symbiosis dysbiosis occurs because of the imbalance of microbiome. So this microbiome analysis can be done using next generation sequencing, where one can go for targeted metagenomic analysis, as I mentioned previously, using 16S ribosomal RNA gene sequencing, or one can go for shorter metagenomic analysis. So in this situation, if you look at inflammatory bowel disease, you have the main arm of this entire inflammatory bowel disease is the dysbiosis of gut microbiota. And that has an interplay with lot many host factors in the diet, and that leads to inflammatory bowel disease. And to further pinpoint where are the problems, you look at this interesting picture where you have a healthy microbiota, where you have high bacterial diversity and less codovirals richness, high fungal diversity. This leads to immune tolerance and mucosal homeostasis, and there is no IBD. And as and when you have less bacterial diversity, and then you have more codovirals richness, less fungal diversity, it leads to inflammatory response and mucosal ulceration, and which is associated with IBD. So this is the impact of analyzing the gut microbiome with respect to various diseases. So I think with these, what we have probably understood that next generation sequencing has a major role to play with respect to infectious disease surveillance. And if we talk about viral diseases, and then other than that, then we have a special disease like tuberculosis, where it has got a role to play, especially for diagnosis and detecting the drug-resistant mutations. And finally, and then also very important role to which is there in surveillance, detection, surveillance of antimicrobial resistance. And lastly, in the case of gut microbiome. So I think with this, I would like to thank you all for your listening. And then if you have any questions, I'll be very, very happy to take the questions now. Um, can I stop sharing my slides?
Yes, sir. Thank you so much for such a wonderful talk. And if any of the participants have any questions, please feel free to unmute yourself and Dr. Abhishek will be able to answer. Okay, so if we don't have any questions, I would like to now welcome Mr. Raja Varman Kitu, where he'll talk about virology and help us go through various public repositories and understand various data sets. Mr. Raja Varman Kitu has completed his Master's in Bioinformatics from Shanmukta Arts, Science, Technology and Research Academy, and his expertise lies in bioinformatics, computational biology, genomics, NGS, comparative genomics, and cancer genomics. He is working as a senior technical officer at Kokila Bhentirubhai Ambani Hospital as well. Now, I would like to welcome Mr. Raja Varman Kitu. Over to you, sir.
Yeah, thank you so much for Biotech for giving this chance. And I think Abhishek have set up a stage for me, where, like explaining all the basic things. Can you just confirm if you can see my screen?
So it's coming up. Yes, I can see your screen now.
Yes. So my name is Raja Varman. So I'm working in Premas Life Sciences as a bioinformatics specialist. So today we are going to speak about, probably we are going to look into what are the databases which is relevant to virology. Specifically, we are going to see. Yeah, so this is our agenda. First is we are going to see basic resources of SARS-CoV-2, which has been the brief. Yes. You can hear me, right?
Yes, sir, we can hear you. I think
Yeah,
that was a disturbance in between.
No problem. Yes. So there are two things which you are going to see. Thank you so much. Yes. So, yes. Yeah. Yeah, I have. So two topics we are going to see. First is basic resources of SARS-CoV-2. Another one is infectious world as a eagle eye. So what is the basic resources? First of all, when it comes COVID, there are two things which we need to look into. One is a global perspective, another one is local. When I say global, we need to know, again, I'll be speaking mostly on the repositories today because that is what we plan, right? So when I say repository, probably the raw data which will be put up in the NCBI SRA site, and then the final output probably goes in this site. And then what are the annotation softwares we'll be using, or probably the databases we consider. And then in India particularly, how does the summation as well as the data sources are placed in? The first thing is SRA. SRA, we all know, probably most of the people who are in the NGS domain knows all the raw data of NGS goes in SRA. So SRA has, you name it, any kind of NGS data, it has, right? So it's simple. You will be creating an account and then you need to fill a form and then you need to submit your sequence. You'll generate a ID on it. So that's how the SRA works. Again, SRA is basically from, as you can see, from NIH. So it not only has a raw data of NGS, apart from that, it has a variant databases, genetic test related, again, variant like clinically relevant variant, normal variants, and then there is so it has multiple resources in one place. These are all relevant to medical genetics. And so it has, I would say probably end to end. You name it, any file formats related to NGS it is having, plus you name it any particular kind of studies, you can get those details, in the sense including the literature survey, publications, clinical trials, almost everything. So how does the SRA works in general? For the submission, people generally go for, as I told before, you will be creating a login and then you will be submitting. As a user, if you see, you need to create an account, you need to locate the data set, and then you need to filter it, and you will be downloading. Again, there are multiple like ways you can download the resources. And then finally, you'll be converting the file. Then you're ready to go for the analysis. So how do I locate my data set? That is the first point. So as you can see here, this is an NCBI page and this is a SRA tab. So I just typed the virus, probably the name of the content which you want to look into. In our case, it's like COVID. So the moment I type in whatever the submission so far has been done with respect to this study, it has been listed. As you can see, there are this many lists have been this many numbers has been seen. The left side, if you see, the possible filtrations are available. The more and more filter we apply based on the type, and then based on the libraries, based on the platforms, and then what kind of data set you want to have it, and like say probably the resources as well. Probably this is from AWS, right? And what kind of file types you want to look into. So based on the filtration we apply, right, we'll be end up with the relevant, probably less number of data set which we need. So that's how we locate and filter. Second is, we can't use the data which is straight away, which is put up in the SRA. We need to go for a tool called FastQ dump, which will be converting the SRA file formats to a FastQ file, which can be used for the, like say, the further proceeding. So that has been done by this particular kind of commands. So once it has been converted, the file will be in this kind of format. Again, this is a typical FastQ file with the header sequence, and then the kind of same header, and then the quality values, right? So that's what about the SRA. So again, whatever we discussed, it's not only for COVID. For name it any kind of NGS dataset, that is the same procedure remains, like say same. Next is we are going to see GISAID, very specific to COVID. What is GISAID? We almost know, I think even Abhishek have covered very nicely. So GISAID is basically a submission database where, again, we need to fill up a form. We need to give all the details about the like source of the sample, where it is processed, which chemistry is being used, which machine is being used. You'll be giving all the background details of the data set, and then you'll be giving a final data. Final data, when I mean it's a FASTA, right? So how does the whole thing works basically? This side, when you submit, it records, and then this particular diagram, which says very clearly is from the day COVID started to still today, how things were being reported. Particularly when I say things, it means FASTA will be getting loaded in their repository. So where does it start? What is its origin? Almost we can figure out the source of the sample set, as well as how faster the sample sets were growing across the globe. So this particular plot, in fact, it's a visual plot. I'm just showing a snap. It's a visual plots which start from the day one of COVID, which been reported to probably CDC to still today. how much cases getting reported. So it's basically a trafficking plot which tells very clearly what is the origin and where do we stand at this point. Second, this is again, it's not just the repository basically segregated in the continent and country wise. Again, when it comes India, the major player is basically INSOCOG, which has been already told before. So it has almost all the individual country segmentation. Our case, it is INSOCOG. Let's see. Further, I'll be discussing more on that. When it comes referencing, this is a probable references which we need to look into when it speak about this site. How does it functions and what are the key points it has? Next, I'm switching to a next clade. This is mostly for annotation purpose. What does it does? Again, we'll be submitting a consensus sequences here. When I mean consensus, it means the raw data which we generate, probably either be it a surveillance or for even the normal sets. When you go for sequencing, you obviously mapping with the OGAN reference, and then you'll be getting a probably the BAM file, and then the BAM will be converted to VCF. You'll be getting all the variant calling files, and that will be moved to like consensus FASTA file. Those consensus FASTA files will be moved to Nextclade to make sure. So basically, it's like a blast search, which generally we do using a simple sequences to map and check what are the sequences, like it's hitting with what are the sequences. Same way here with respect to COVID. Globally, what has been reported so far will be going for that kind of hit. So it's basically it's our annotation databases which takes a given individual samples and it annotates and gives says which classification it belongs to. So the Nextclade, how does it defines? So as you can see here, it says the origin, the newer groups, whatever is being let's say formed or probably whichever the classification it belongs to, it gives a very clear. Currently, we somewhere stands somewhere here. So in that case, you can see what is the origin, what are the branches. So basically it tells all the nodes, subnodes, and the branches which belongs to. But when you put in your own sample, it checks with all possible variants which is there, and then it classifies and it tells very clearly which clade it belongs to. That's what the functionality of this database, right? How does the page looks like? This is a page. Again, there are two options. You can go for a web browser. If people who are not comfortable with CLI, they can straight away put in the consensus FASTA file in this web browser. It gives a clade classification straight away. The other option is you have option for going CLI. You'll be downloading, installing in the command line, and then each time when you you will be calling the updated databases in the background, so it annotates and classifies and gives output, right? So these are all the repository where you'll be getting the resources. Next is Pangolin. Pangolin is basically for lineage classification. This is a page for Pangolin. How does it work? So probably the font size is small. So what does the Pangolin gives is lineage details. So when I submit the data, again, the cases remain same here. I'll be submitting the same consensus file in this particular database. It checks for all possible or previously reported sequences, and it classifies clearly and then says which lineages it belongs to. That's the role. It lists almost globally everything. It's updated each and every minute in that case, right? The important thing to be noted down this is variant of concern, right? So as you can see, according to them, currently the global scale, these are all the 5 variants top list. So the individually, since it's an Indian scenario we are speaking about, the variant which is important with respect to Indian scenario, which is B1617.2, right? So this not only gives a number, rather it gives all kind of segments. How does the number got in cases got increases with respect to time frame? What are the variants are seen? So it gives almost all. So these are all the variants which is responsible for this classification. With respect to countries, how many countries, like country wise, how many cases was being reported? It gives all the description behind this particular, probably the classification, right? So that's what about the COVID databases. So we have seen how do we get the raw data from SRA. We have seen how do we classify them using, like how do we submit the final data which is this side. How do we classify using 2 different databases, right? Which is Nextclade and Pangolin. Finally, we are going to see local. Local, when I meant it means in Indian level, what are the repositories and who are working behind, right? So which has been already told, it's INSOCOG, right? So INSOCOG the repository will be looking like this. Again, here you will be feeding in all the details which is relevant to the sample resource, like say sources of the sample, right? As well as the final outputs. Who are all the in charge at this current, like say this point? Previously, it was just 10 labs. And then later at this stage, since the workload got increased, there are much more labs has been added. These are all the final labs which are all doing the COVID sequencing, right? And reporting in the GSI. And with respect to Indian scenario, what are all the, like say VOC, which is a variant of concerners? These are all the alpha, beta, gamma, delta, omicron. These are all the main things which is being looked into. When it comes to variants of interest for our case, these are all the things. So these are all the things are always in the surveillance tab for us also, right? So when it comes in SOCO community submission part, right? Not only there, you can submit the data even here. It is basically maintained by IGIB. You can see how many genomes has been recorded, how many classification has been done so far, right? And who are all involved in this, like say in the team. So this here you can see the same way decide whatever the classification we saw, it is being put up in the IGIB page as well, repository page, with all the pangolin lineages plus what is the contribution with respect to the time, right? So far what we have seen is a COVID story. COVID story when I means including the databases and the repositories, right? Now we are going to see one of the classical paper which is being discussed. Probably it's an open source tool, which very nicely written with respect to what does the clinical world should follow. So reason for taking this paper is they have covered almost all possible analysis in one go, right? So as you can see here, it covers not only the virology, it has both, it captures the details of host and the pathogen, that is the first key. And they have considered different kind of sample type, as you can see here. Plus, they captured in different time frame. I'm speaking about the data set first, and the treatment option, assay, target. So it has almost all the points which is needed so that we don't miss any single point from the analysis, right? So as I told before, these are all the hosts which is being covered. Nearly 77 virus and then 33 hosts is being captured in one go, right? For the study. So this is one of the good study, probably good repository I would consider for any person who would like to explore all possible NGS solutions in one place, right? So there are 22 different assays. And again, as a technology, if you see, RNA-seq, ChIP-seq, it like say it most even single cell, ribose-seq, CLIP-seq. So it has covered the major things which is being utilized in the microbial field. And when it comes the repository, how do they place it? They place something like this, which is basically individual strain wise, virus wise. When you get inside, you can see the respective data set. So when I say influenza, they have taken a sample from the sample study is based on the lung, and then they have did a RNA-seq. So expression profiling we can get out of this. The same way for individual tab, when you keep on getting like say individual folder or probably the hierarchy wise they are given. So based on your study, you can get the relevant data sets, right? So it's not only they have captured the data set, rather after the analysis in the downstream level, they went to differential expression, gene ontology, KEGG pathway, splice site capturing, right? So they almost captured all important things and they have put up for visualization for the public use, right? So these are all the assays, as you can see, literally all possible things is being captured, right? And sample type, when I meant, it has almost, there are 6 tabs are there. We are seeing only one of it, right? So that many sample types has been captured. Probably the cell type has been captured. And again, tissue types, there are 6 tabs. We are seeing only one of it. So number wise, it's really huge. Data sets are very big. So that when you make a point out of like say any analysis you do, and then when you make a point, like say key points, when it makes sense only when you have this kind of data sets, right? So with respect to the data sets, as you see, the major databases are already in place as a repository. Analysis wise, if you see, almost all kind of analysis is being performed so that you cover all the segments so that we don't miss any information. And it's a huge repository which is based on the MySQL, right? When you do the analysis, as you can see, again, as I told, host virus, so you capture all the angle, probably all the segments. In that way, your outputs are more reliable, and it gives more insights, by the way. That's a key point of the whole like say this particular repository, right? So when I meant eagle view, what does it means? I'm not just looking into one segment and then saying, yeah, this is what happening. Rather, I just get out of the frame and see from very far so that I get an idea of what is the holistic view you're trying to get, right? So here, if you see, the total number of data set is somewhere closer to this, right? And the file format which we are speaking is somewhere like, say, these are all the files we are getting as an output. And as an analysis, like host pathogen interaction, dynamic expression, like say meta virus signatures. So we are capturing all possible details, including the single cell, right? So this kind of the future microbial world should be like multi-omics related projects should be handled so that you can get a holistic picture, and when you make a statement to public and it's more reliable. That's a key point which I'm trying to put it over here, right? And with that, I'm probably closing the talk. If you have any questions, you can shoot.
Thank you so much, sir, for such an insightful talk. Karl Marx, I'll be sharing the recording of the session with you. That will be helpful. And if anyone has any questions, please feel free to ask. Okay, so I think everyone is just taking time to absorb the concepts that we went through today. Okay, so you can either reach out to us on our mail IDs and we'll be able to pass your questions ahead then, if anyone is there. So I would like to hear from our mentors, maybe if they would like to give some highlights or discuss about the session, maybe.
Thank you, Sonalika. I mean, that was wonderful session by both Abhishek and Rajavarman, and we enjoyed a lot. I mean, there was lots and lots of information that I think all the participants are finding it really difficult to digest, and then they will come up with some of the questions. And I hope we will be equipped enough to give them some directions in these in the topics that you have discussed in extensively and also in great detail, and also in a very interesting fashion. I could not account for the last 1.5 hours how it went through. I think I'm in the same state as the participants. I don't have any specific question in effect in detail. I have some of the questions to Rajavarman, if I can shoot him. So
yes, sure.
Yeah, he spoke about some of the file conversion and how to take links from SRA and then convert into FASTQ files and etc. So these conversion, will they work on a normal computer, or do you need high performance computing for this?
So
is the space only is the limitation is only on the space, or is also on the processing power also?
Yes, it's a very good question. So when it comes databases, since we are speaking about small organism at current, like say this point, so server is not compulsory. Even the high-end workstation can do. What I meant is 2 TB storage with probably 64 GB RAM is fair enough to start with. But if the high-end machines, and then you are going for bulk number of sequencing, probably like say huge number of samples, then probably the server should come in picture. So when it comes conversion part, yes, it can done in the simple workstation also and the server also. There's no deal. Only thing is, when the data size is big and then more samples, obviously, I don't recommend to do in the smaller configuration. Rather, it's better to do in the server. Yes.
Yeah, perfect. Thank you very much, Raja. Thank you.
Yeah.
And I think that will be it. That's an excellent an overview of what are the different tools that are available, and it tells importance, and it tells data that we have within our reach, right? All these data is they are not hidden somewhere in anywhere.
Yes.
It's there.
Yeah. So yes.
Yeah.
So the important is people generally think, no, I have not seen NGS data. I don't know where to get. Plus, where do I start, right? So
exactly,
so in that case, yeah. So that's the main intention, where it's always out. You just find it in the right place. That's it.
Excellent. Thank you very much. You made sure that everybody got through this point, and we are wiser than what we were 1.5 hours
Yes.
ago.
Yes.
You. If Dr. Mohit has anything else to add, or Sonalika has anything else to summarize.
Thank you so much, Dr. Raghav. It was actually a very wonderful session, and it was very insightful. And I would like to thank Dr. Abhishek and Mr. Rajavarman for taking out the time for this session and brainstorming with us and letting us know about so many fun facts and things around. So thank you so much, sir. And we'll be looking forward to having great sessions with you in future as well. And as a part of the symposium workshops, we have a session in March 2 with Prema's Life Sciences, and we'll be sharing the details of the same with all of you very soon. So looking forward to meeting you all in our sessions ahead. Thank you for joining. And if you have any questions, please feel free to reach out to us at marketing@omicslogic.com. Let me put the mail ID in the chat box. And you can also reach out to us at support@pine.bio, and we'll be there to assist you. Please make sure that you have added both of these mail IDs to your contact list or email address book, so that none of the mails drop in your spam box or promotions box. Thank you everyone for joining. We'll be looking forward to meeting you once again. Bye.
Yeah. Thanks a lot. Thanks.